my presentation is the bioinformatic analysis of genome evolution in trees. I'm based here in CLC Bio, supervised by Annika, but I also have a supervisor in Queen Mary University in London. So there are three general themes to my project, so I'll go through them one by one. So firstly, conservation of ash trees relative to the ash dieback fungus. Secondly, hybridization in two species of birch trees. And thirdly, some epigenetics as well. So start off with ash dieback. This is the fungus, the species name is Hymeноcyphus pseudo-albidus, but it's also known by this name as well, Cholera fraxinia, which is kind of easier to say, so it's a bit better. But basically, the fungus, you can see it on the stems here, the stems of the leaves. They have these spores. And basically, it ends up killing the tree if it gets infected in about a few years by killing the leaves, and you can see that kind of goes a bit droopy. And as of last week on Monday, there were 523 confirmed sites of infection in the UK, which you can see by all the red dots on the map over there. But this is an infection that actually started in Poland in the 90s, and it spread all throughout Europe, and it came to the UK only last year, so it's really quite a recent spread, but really quite quick. And it's actually been quite a lot in the media as well, so the public are really quite aware of this problem that we're having. So these are some examples of some news headlines. And actually our supervisor and Yasmin were on the BBC News last week as well. So that was very cool. So it's getting a lot of media attention. And there's also this app you can get on your phone, which I actually downloaded last week too, called Hashtag, and it's where if you see an infected tree, you can plot it on your phone, and then someone goes out and looks at the infected site to see if it is really infected or not. So basically this is something that the public in the UK are really quite aware about. So there are approximately 90 million ash trees in the UK. In Denmark, about 99% of the ash trees were infected by this fungus. So it's kind of estimated that the same percentage is at risk in the UK. So this could be a potential loss of 89 million trees, which is really, really bad. There's a lot of ecosystems that are kind of dependent on this tree, so it's pretty bad. But luckily, there is some natural genetic resistance in the UK population, about 1 or 2%. So what we want to achieve, or the scientific community wants to achieve, is to breed a population of these naturally genetic resistant trees so that we can conserve the ash trees without having to use chemicals like fungicides or something like that. But to do that, to look at the genetics, we first need a reference genome sequence, so a basis, like a baseline to compare other gene sequences to. So that is what our project is doing. And Yasmin has extracted DNA from a selfed tree, so like a parent that was mated with itself. So Yasmin has extracted DNA from that. We sent the samples off to Germany for Eurofins sequencing, and that was using Illumina and Roche 454 technology. So we have like a hybrid, two types of data. And then I have been busy assembling the reads into genome sequence using our own software that we have here, the CLC Genomics Workbench. And we're also getting some transcriptome and also maybe some methylomes, the methylated DNA data soon as well, so then we can put them all together. And then what we want to achieve is to publish this assembly and all the different types of data, and so the scientific community can use it for looking for resistance genes, for example. So this is the general method by which I assembled the reads. So we get the raw reads, and then quality trim them to a minimum Phred score of 15. A Phred score is idea of the quality. So the higher the Phred score, the better the quality. And then I did the step merging overlapping reads, and this is explained here. So in some of our libraries, we have a 200 base pair fragment of DNA, and that's what we're sequencing. And we're using paired reads, so we have one read coming from this end, and the other read coming from the other end. And these are 100 base pair reads. So what should happen is that the coverage is equal along the entire length of the insert DNA, and they should kind of meet in the middle. But some of these 200 base pair insert sizes aren't actually 200 base pairs. Some of them are bigger, some of them are a bit smaller. So if you get a smaller insert DNA, so say 150 base pairs, your reads are still 100 base pairs. So they end up overlapping by 50 nucleotides in the middle. And then you can actually merge these reads together and just get one long read, which kind of helps by reducing the amount of data that you have and reducing the complexity of the de novo assembly algorithm. So it kind of helps. So after you do that, then you can just do a de novo assembly of all the reads, and then you adjust the parameters, change some things, and kind of do it again. And I've ended up doing this maybe about like 20 times so far. So it's like a continuous process of just changing and looking at just de novo assembly and comparing them. So we had a version of the assembly that went up a few months ago. We've actually just updated it last week and we published a new version, which is on this website at ashgenome.org. It's currently about 280,000 quantigs. The total size was 1.4 gigabases, which is slightly more than the genome, which is 950 megabases. So it's slightly assembled more than it should. And this N50 is a measure of the how fragmented the assembly is. So, for example, this is the percentage of the assembly, the genome, and then you go on the 50%, and then this is the contig size. So this is like 14,000 here. So it's like the median position of the contig. And it's kind of used as a measure of the quality of the de novo assembly, so a higher N50 is kind of better. But I've actually, in the last few days, I've managed to get this number down and this number down, so it's continuously improving. So we're on to birch hybridization. So in the UK, there are these two species of birch trees. First one is downy birch, which is quite a big tree, 20 meters, but it's a tetraploid, so it has 4 sets of chromosomes, so it has 56 individual chromosomes, and the genome size is 880 megabases. And then we have this other birch species, the dwarf birch, Betchulinana, which is a little shrub, only a meter high, found in Scotland and other places. And it's diploid, so it only just has 2 sets of chromosomes, so it has 28. And genome size is just about half of this one, so 450 megabases. And these have hybridized together to make these hybrids. And the height really quite varies a lot, so some of them resemble more the tree, and some of them resemble more the shrub. And the ploidy also varies, but most of them are triploid, so they have 3 sets of chromosomes. So it's kind of an intermediate. So in terms of the gene flow or contribution of the DNA going into the hybrid, obviously we have contributions from the 2 parents going into the hybrid. But these hybrids can then interbreed back with the parents because they're so close. So we end up getting gene flow back to the parents as well. And then, because this happens continuously, you end up getting gene flow between the 2 parents. So what we want to do is using this RAD sequencing, restriction site associated DNA sequencing, to analyze the mixing or integration of different genes in the parents. So what the RAD sequencing is sequencing regions around the restriction sites. So for example, if you have this DNA and restriction sites here, you end up getting the reads stacked up like this. And this is a restriction enzyme that we use to cut at this cut here. And then what we want to do is identify markers such as SNPs that are specific to each of the parent species, and then we can identify these in the hybrid and look at the levels of integration that's happening. And we have data from about 120 individuals of mixing of the different species. So epigenetics is covalent reversible modifications to the DNA. These are responsible for altering gene expression. So, for example, methylation of promoter regions is linked to gene silencing. And these are influenced by our environment, and they're reversible. They can be inherited by the next generation as well, which is interesting for us because you can also get induced defense, like resistance in plants, and then this can be passed on to the next generation, which could be useful for us making a breeding population of resistant trees. So what I kind of want to do is looking at linked epigenetic patterns. So these are patterns that happen that are associated with each other. So for example, like a missing methylation region here and the methylated position here, and those are kind of linked together. And these can form a haplotype, so for example, a resistance haplotype or something. So I want to kind of make a method for identifying those as well. So the overall aims, analysing gene introgression and hybridisation in the 2 birch species and the hybrids. And I also want to develop a workflow for analysing this type of data in the workbench, because it is possible, but it's not entirely clear in the workbench at the moment. Make an ash tree genome and publish that. Develop a tool in the genomics workbench for looking at methylation data. So, for example, bisulfite sequencing is the sequencing method for doing that. And then do this idea about looking at linked epigenetic variants as well. Stop yeah. So my areas of expertise in general, like the biology,